Hello students, in this video we are going to continue on in the 1.2 notes. Specifically, we're going to look at topic 2 in this video. But in these notes in unit 1.2, we're looking at these different types of macromolecules that are found in living things. There's 4 main types of macromolecules that we're ultimately talking about. We already talked about carbohydrates in topic 1, but today we're going to look at proteins. Now this is probably going to be the longest video, mostly because of these notes, because it has a lot of information here about proteins you guys need to know. So bear with me. We'll try to go through it pretty quickly, but efficiently in detail. So anyway, just like carbohydrates, what we're looking at is these giant molecules found in living things that are ultimately made up of these repeating subunits called monomers that form all these huge variety of structures that do different things in your body. So proteins, this is a different class of macromolecules. They have a huge variety of structures that they take on with a huge variety of functions that they're accomplishing inside of your body and inside living things. So proteins basically, if I had to summarize it, they're basically doing almost everything inside of you. Proteins are responsible for a huge, unimaginable number of tasks and things that are happening inside of you and inside of cells. They do a lot. And at the end of this video, we'll kind of talk about some of the different types of proteins and generally what they're doing, but ultimately you're going to see they basically are doing a lot. And really proteins are controlling and responsible for everything that's happening. They're the ones actually accomplishing all these different things happening in cells, and ultimately are responsible for all your traits and characteristics as well. And we'll kind of get into this a little bit more later on, but like, for example, the fact that you guys have green eyes or brown eyes or blue eyes, like that trait is because of proteins. There's a certain protein that's being made in your cells in your eyes that are causing a certain pigment to be produced. Same thing with why you have curly hair or straight hair or wavy hair. It's the type of proteins that you're making in your hair that are responsible for that. So they're doing a lot of different things and responsible ultimately for the different characteristics and traits that organisms have, along with the millions of things that they're doing inside of the cells. But, so let's get into the structure of proteins then. Like I said, these are polymers, meaning that they're made up of repeating subunits. The repeating subunits that make up proteins are called amino acids. And those amino acids, which are the monomer, are going to bond to each other over and over and over and over again to build a polymer. And we call that polymer a polypeptide. So polypeptide is the name of the polymer for proteins, and amino acids are the monomers. Just like for carbohydrates, just to remind you, in carbohydrates, there's monomers called monosaccharides that bind together and form polysaccharides. While in proteins, there's amino acids that bind together to form polypeptides. The elements you're going to see in proteins, and on these slides, actually, you'll see a lot of these guys these are a little bit different than the ones in carbohydrates. In carbohydrates, just to remind you, there's carbon, hydrogen, and oxygen, and that's basically it. In proteins, there's carbon, hydrogen, and oxygen, but also we're going to see some nitrogen and some sulfur as well. So these 5 different elements are what you're going to find in proteins. So let's take a closer look at amino acids. These are the building blocks, the monomers that create proteins and these polypeptides. There's 20 different amino acids that exist. You guys don't need to, I'll show you them in a second. You don't need to memorize them or know their names. They each have different names, but they all have basically the same overall structure. So in an amino acid, every amino acid is going to have an amino group, which is this guy, a carboxyl group, and something called an R group, all attached to a central carbon, which we call the alpha carbon or the central carbon. So attached to the central carbon is an amino group, and then there's a carboxyl group, and then there's something called an R group. This amino group and carboxyl group, we actually looked at those in the unit 1.1 notes when we talked a little bit about organic chemistry. These are some of those functional groups that I pointed out, and you guys do need to know these. You need to be able to recognize that this is the amino group, this is the carboxyl group, and those 2 groups are found in amino acids. And then there's something called an R group. Now this R group, R doesn't stand for an element. There's no element R. It actually stands for a variety of different structures you could find here. And so it's this R group that's actually, which we call a variable side chain. or just side chain, or just R group, whatever you want to call it. This is what makes all 20 amino acids different. So all 20 amino acids have an amino group, a carboxyl group, a central carbon, and then it's this R group that's different. So if you look at this table here, which I'll kind of move myself here, on this table, you're going to see the 20 different amino acids that I was talking about, and here's all their names. But if you notice, they're all basically the same. They have a central carbon, amino group, carboxyl group, central carbon, amino group, carboxyl group, central carbon, amino group, carboxyl group. What's different about them is this highlighted region called the R group. This is the R group of different amino acids. Every amino acid has a different R group. And so there's 20 different amino acids because there's 20 different R groups you can find attached to these amino acids. Now these R groups are going to give these amino acids some different properties. There's some R groups that are hydrophobic. They don't like water. There's some R groups that are hydrophilic. They do like water, and they're polar and like water. And then there's some that are actually ionic. They're ionized. They actually have a charge, a negative or positive charge. You can kind of see closely if you zoom in on this table. This one has a negative charge, a positive charge, a negative charge, a positive charge, a positive charge. And so these actually have fully charged regions on them. They're ionic R groups. Ionic R groups are hydrophilic, and polar R groups are hydrophilic. They both like water. So these guys and these guys, the bluish ones, they like water. So just know that there's certain R groups that are polar or ionic, and they're hydrophilic. And so these amino acids like water, like being around water. Now these ones up here, what you're going to mainly see in these guys, the non-polar ones, is that their R group is just consisting of hydrocarbons, like carbons and hydrogens, 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 carbons and hydrogens. Even in this little fancy ring, this is a bunch of carbons and hydrogens that they're not showing you in this summarized structure. And if you guys remember what I told you in a previous video, is that hydrocarbons are by default non-polar, hydrophobic. They don't like water. It's like this doesn't like water, this doesn't like water, doesn't like water, doesn't like water. Like these hydrocarbon containing or dominant R groups. They don't like water. So that's how you can kind of tell if they're going to not like water or not. If you look at their R groups and see if they're just a bunch of hydrocarbons, they're going to be non-polar, and they're going to be hydrophobic. And if you see some other special groups, like some like these guys, they're gonna be hydrophilic or more polar and like water. Again, you guys, please don't memorize all of these, but you do need to be able to look at an amino acid. There's an example of this in your notes on the left side where you have to look at, if I showed you an amino acid, you should be able to find the R group and maybe predict if it's going to be non-polar or polar or hydrophobic or hydrophilic based off that R group. Like this one, if I looked at this R group right here for leucine, and that's the name of this amino acid, I found its R group. If I look at this R group, it's just a bunch of hydrocarbons. So I would know that this is a non-polar amino acid, which is hydrophobic. It's not going to like water. Anyway, those are the different amino acids. Now, amino acids, these are the monomers, and they're going to bind together over and over and over and over again to form polymers, which are ultimately called polypeptides. And so these amino acids are linked together through dehydration synthesis reactions, which we've mentioned multiple times already in previous videos. And that is the type of reaction that links our bonds, monomers together, and results in the production of a water molecule that's going to be produced and released as this reaction is taking place. And so that's going to bond this amino acid with the next amino acid and with other amino acids. Now, this bond that's created, the name of this bond is called a peptide bond. That's the bond that connects amino acids with other amino acids. So this is a covalent bond called a peptide bond that connects this amino acid to the next amino acid and the next one. You can see these peptide bonds forming through these dehydration synthesis reactions. Now, the peptide bond, specifically, if you look here, it's between the carbon and the carboxyl group of this amino acid and the nitrogen and the amino group of this amino acid. That's what's happening here. That's where that bond's going to form between this carbon and the carboxyl group and this nitrogen and the amino group between these two different amino acids. And you do have to be able to point that out. Now in this picture, it's kind of easy to point out because they highlighted everything. So you can look at this and you're like, well, that's the peptide bond joining this piece with this piece because they're shaded. You can tell they're two different amino acids. But even if they're not shaded, you guys need to be able to find the peptide bond. So imagine there was no like yellow, green, purplish shading on here and it was just a bunch of atoms linked together. You got to be able to point out a peptide bond. And if you look carefully, you can kind of see where the R groups are, these special side chains. And then you can see the amino group, the essential carbon, which is going to be attached to the R group, and then the carboxyl group. and then you'll see another amino group, essential carbon attached to the R group, and the carboxyl group. And so that bond, you kind of have to be able to recognize the difference between this amino acid and that one. That's the bond we're looking for. Some students confuse it with this bond right here. Like this is a bond between carbon and nitrogen, just like the peptide bond. But this is just the essential carbon attached to the amino group in the same amino acid. And so that's a covalent bond, but that's not what we call a peptide bond. This is a peptide bond, like right here and like right here. Anyway, when these are linked together, you start forming polypeptides, which are going to contain lots of these, sometimes hundreds of them, linked together. And the two ends, there's going to be two different ends. There's going to be an end that contains an amino group, and then all the way on the other side of all these amino acids being linked together, in this picture there's 3 of them, but on the other end, even if there is 100 amino acids, there's going to be a carboxyl group. So one end is going to have an amino group, one end is going to have a carboxyl group. We call this the amino terminus, and we call this the carboxyl terminus, or the N terminus and the C terminus. There's 2 different ends. Now when we have polymers that have 2 different ends that are chemically different, we say that it has directionality. We say that proteins or polypeptides are directional molecules because one end is different than the other end. They have 2 different chemical ends. Like I said, you can see this end doesn't look the same as this end, 2 different ends. So you guys do need to know that there's 2 ends on a polypeptide, the N terminus and the C terminus. And this is going to be important because if I want to add more amino acids to this, the amino acids that I want to add to this they can only be added to one of these ends. And so if I want to continue building on, in the cell, if they want to continue building on this polypeptide and adding more amino acids, they're going to have to be added to the carboxyl terminus. That's the end where new amino acids will be added to this end right here. Or in this picture up here, it'd be this end right here. So if I want to add a 4th amino acid to these 3 amino acids, it's going to be linked on this side, not on this side. They can't be joined to the N terminus, always on the carboxyl terminus. And so that's kind of, some of that is summarized here. You guys can see a lot of that amino acids being joined together, and then you end up with a whole chain of amino acids. These can get very, very long. All of these are amino acids with different R groups, and there's always going to be an amino terminus and a carboxyl terminus on the 2 ends. They're going to be different. So now let's talk about maybe the most important thing here in the notes is the ultimate structure that a protein takes on. And so as you link these amino acids together and start building these long polypeptides, ultimately it's going to take on a 3-dimensional shape, which we call a conformation. A conformation is just a fancy word for its shape, its 3-dimensional structure. And so proteins, this is really important. Their 3-dimensional structure, their shape is ultimately what's going to determine and affect their function. And so these proteins have a bunch of different jobs to do inside your cells. And those jobs that they have and what they're doing is going to be based off of their structure. So depending on what structure they take on, they're going to have a certain function, which is a common theme we're going to see throughout all 4 of these topics. The structure is what affects its function or determines its function. Now in proteins, we actually kind of can look at 4 different levels of structure that exist in a protein. And this is where it gets maybe the most kind of complicated or detailed. And so we're gonna look at each of these levels on the next few slides and kind of look at the difference here. But basically what we're gonna see is there's gonna be a sequence of amino acids, and they're gonna start folding up into these different structures and shapes that are gonna continue folding up into an overall structure, and you're gonna end up with this final 3-dimensional shape that the protein's gonna take on. And then it will now be able to accomplish certain tasks inside of your cells. And so let's start with the primary level. The primary level of a protein structure, so when I say the primary structure of a protein, what we're referring to is what's the sequence of amino acids. So first and foremost, to have a protein, you have to have a polypeptide, a string of amino acids, and there's 20 different amino acids. And so what amino acids are linked together, and in what order are they linked together? That sequence is called the primary level of a protein structure. So here is an example of a protein called Transthyrotin. And this protein actually consists of over 125 amino acids. And here's all 125 amino acids and what order they're linked together. This is the primary structure of this protein. So this is ultimately where it starts. You got to put amino acids together. They're putting there's certain amino acids that we're putting together in a specific order, and that's level 1 here, okay? And changing this amino acid sequence is ultimately, if you change some of these amino acids or have completely different amino acids in a completely different order, that's ultimately going to affect its overall structure, which you'll see in a moment. But anyway, that's the primary level. Now the secondary level, one of the things that are going to happen after these amino acids have been put together is they're going to form these, they're going to start folding into these 2 different specific structures that we commonly see in proteins called alpha helixes and beta pleated sheets. So like these helical structures and these zigzag folded structures. In most proteins we find these guys somewhere in the polypeptide as it's folding up. So you can see like this long chain of amino acids, this polypeptide. It's starting to fold up. Some regions are starting to form these helixes and some regions are starting to form these zigzag sheets. And that's because of hydrogen bonding that's occurring between these amino acids. So in some amino acids, when they're next to each other, they start forming a certain pattern of hydrogen bonding that leads to this zigzag folding, these beta pleated sheets. And sometimes they are next to other amino acids where they form a slightly different pattern of hydrogen bonding between each other that forms these alpha helixes. So this is because of hydrogen bonding between these amino acids that start forming these alpha helixes and beta pleated sheets in this polypeptide that's starting to fold up. And then we get to the tertiary level of a protein structure. And this is when the protein is going to fold into this is when the polypeptide, the string of amino acids, after it's now folded into its the alpha helixes and beta pleated sheets, it's going to continue to fold in on itself into a specific globular, which just means 3-dimensional shape, that it's going to finally take on. So you guys can see this long ribbon, which is just representing this sequence of amino acids that was put together. This really long sequence of probably hundreds of amino acids. Sometimes there's alpha helixes that formed and beta pleated sheets. That's the secondary structure. But look at how this ended up folding on top of itself into this final globular purple blob here, this shape here. And that's because of the final folding that's taking place. Like this guy's going to fold backwards like this, and then loop around like this, and then fold this way, and then this guy's going to fold back that way. That final folding that's taking place, that's the tertiary level of a protein structure. And this is because the reason it's folding like this and why it's folding into this specific shape is because of interactions between the side chains, which are these R groups. So these R groups now that all these amino acids have different R groups, they're going to start finding other R groups that they can form certain interactions with. Like some R groups can interact with other R groups and form hydrogen bonds. And so like this amino acid has this R group right here, and then like 50 amino acids later, somewhere way over here, there's another R group that has, there's another amino acid that has this R group. And those 2 R groups find each other and they can form a hydrogen bond. So it causes this ribbon or this string of amino acids to fold in like this because they're now interacting with each other. That's one type of interaction. Some of the R groups hydrogen bond with each other. Some of them can ionic bond with each other. I pointed out earlier how some amino acids, they have an ionic R group, an R group that's positively charged, and there's some R groups that are negatively charged. So this amino acid where my mouse is pointed, this amino acid has an R group that's positively charged, and then like 20 or 30 amino acids later, there's another amino acid, and this guy has a negatively charged R group, and they find each other, and they fold in to ionic bond with each other. Oh, fire alarm. They're testing it. 1 second. Okay, yeah, I survived the fire. Sorry, they've been testing the fire alarms all day. I thought they stopped, but we'll see if it happens again. Anyway, so I was pointing out how some of the R groups can ionic bond with each other, so it causes this chain, this string of amino acids to fold in like this so that they can find each other in ionic bond. Some of them are hydrophobic. Like I said earlier, some R groups are hydrophobic, and there's other R groups that are hydrophobic, and they're going to find each other. Like this one over here and this one over here they're going to find each other and be next to each other because hydrophobic things like other hydrophobic things, and that causes this to twist up like this and fold that way. And then sometimes there's even these covalent bonds that can form between certain R groups, like this R group right here and this R group way over here on the string of amino acids. They found each other and they're forming a covalent bond with each other. This is a special type of covalent bond we call the disulfide bridge, which can occur between an R group that has a sulfur and another R group that has a sulfur atom, they form this special covalent bond called the disulfide bridge. This is special because this is really strong. A covalent bond is really strong, if you guys remember. The only way you could break this is through a chemical reaction. Whereas these other interactions, like hydrogen bonds or ionic bonds or hydrophobic interactions, those can easily be broken. They're not super stable. But this guy's really stable, so this helps to kind of lock in certain parts of the protein structure. But anyway, these R group interactions and these R groups finding other R groups and doing these different things with those R groups is ultimately what's causing this final folding of this protein's globular three-dimensional shape. And so for a lot of proteins, that's basically it. There's the primary, secondary, and tertiary level, and then that protein has now folded up into its shape, and then now that it has that shape, it can now go do its job, and that protein is ready to go do its specific task that it needs to accomplish inside the cell. But for some proteins, there's actually a fourth level, a quaternary level. And, oh, I'm sorry, before I get to the quaternary level, there's one last thing I want to say about the tertiary level. These three-dimensional proteins, another thing that we're going to commonly see in a three-dimensional protein is that the hydrophobic amino acids, so the amino acids that have hydrophobic R groups, they're usually going to fold into the inside of a protein, like somewhere in the inside. These non-polar hydrophobic R groups are going to fold inward, and the hydrophilic ones, the polar ones, are usually going to be found on the outside of the protein. And that's because these proteins exist inside of our bodies and inside of our cells, which are made of mostly water. So there's a bunch of water everywhere. Inside a cell, there's a bunch of water. Inside your living body tissue, there's a bunch of water. And so the hydrophobic ones find a way to fold inward to get away from the water, and the hydrophilic ones like to be on the outside. And so that also contributes to the folding of the protein's tertiary level, these hydrophobic, hydrophilic interactions, and where these guys end up because of water. It's going to also contribute to the final shape that a polypeptide takes on. And so in the end, after your tertiary level, you have this string of amino acids that folded into this final three-dimensional shape. Now, like I said, some proteins have a fourth level, and in the fourth level, we call that the quaternary level. And this level, this only applies to certain proteins, a lot of proteins, but not all proteins, certain proteins. And this is when there's actually some proteins that are not made up of just one polypeptide that folded up into a shape, but it's actually made up of multiple different polypeptides that each folded up into a globular shape, and then they all come together to form one functional protein. And that protein functions as one single protein, but it's actually made up of two or more different strings of amino acids, different polypeptides. And we call each of those polypeptides that make up that final protein a subunit. So this is hemoglobin. It's a great example. Hemoglobin is made up, this is the protein that carries oxygen in your guys' blood, found in your guys' red blood cells. And this protein is actually made up of 4 different polypeptide subunits, which means that there was a string of amino acids that folded up into this shape, and a separate string of amino acids that folded up into this shape, and a separate string of amino acids that folded up into this shape and this shape, and they all come together and function as one protein. And so sometimes, like this protein right here is made up of not one polypeptide that folded up, but multiple polypeptides that folded up. And so anytime you have a protein that's made of 2 or more, like in this case there's 4 different ones, we say that that protein has a quaternary level. And again, this is only certain proteins. Collagen is another example. It has 3 polypeptides that come together and form one functional protein. This is another one. The transthyrotin protein is actually made up of 4 different subunits that come together and function as one protein. But again, for a lot of proteins, they don't have that. A lot of proteins, they just are one string of amino acids that folded up, and then they're ready to do their function. Okay. Anyway, those are the 4 levels of protein structure. I kind of want to summarize that in case a lot of that went over your head or that's confusing you. So I have an example here with like these pipe cleaners, I think are a great example to kind of show what all this means. So let's say like this purple pipe cleaner represents a string of amino acids. Let's say it's 150 amino, I don't know, it's like, has a lot of amino acids all linked together, right? And that sequence of amino acids, the amino acid here, 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 that's the primary structure. That's level one. That's the primary structure. First and foremost, you got to put amino acids together, and there's specific amino acids you're putting together in a specific order. It's the primary structure. Now what's going to happen is certain regions of this are going to now start hydrogen bonding in a certain pattern that causes alpha helixes to form and beta pleated sheets. So let's say over here, these amino acids start to hydrogen bond with each other, and it creates these alpha helixes, which I'm just going to show like a little spiraling right there. And let's say these amino acids over here, they start to hydrogen bond with each other, and it creates this type of folding, these beta pleated sheets, these zigzags, like that. So you got some beta pleated sheets right here, some alpha helixes right here. And let's say over here on this end, this other end, there's some more hydrogen bonding that's going to occur that creates more alpha helixes. So let's say there's some more spiraling happening over here. Right, so now we have the secondary structure. There's some alpha helixes that are forming over here and in this area, and there's some beta pleated sheets, right? That's the secondary structure. And then what's going to happen is this thing is going to start folding in on itself because certain R groups are going to find other R groups, and there's going to be certain R groups interacting and ionic bonding or hydrogen bonding with each other, or this one's hydrophobic and this guy over here is hydrophobic and they like to be next to each other. And then maybe this one up here is ionic, and there's another ionic one over here, and so this is going to fold in like this. And so those R groups are going to start finding each other and doing different things with each other, and that's going to create a final, let's just say it's this mess right here, this final globular shape. This is the tertiary structure now, after it's now folded into itself on its, this is its final three-dimensional shape because of those R groups interacting with one another and interacting with water around it. And so now let's say that this protein I'm making actually has a quaternary level. So if it has a quaternary level, what that means is that this final protein is not done. So sometimes this is it. Like sometimes for some proteins, this is it. It's ready to go do a certain function inside the cell. But a lot of times, or not a lot, but commonly, there's some proteins that have another level of structure called a quaternary level. And that's when you're going to have multiple polypeptides that folded up into their shape, this guy folded up into his shape, and then this guy folded up into his shape here. And so now I have three different polypeptides, three different sequences of amino acids that folded up into its secondary and tertiary structure. And they're going to actually come together and interact with one another and form one final functional protein. So now this protein is the completed protein. This is a protein that has a quaternary level because it's made up of multiple polypeptides, subunits, that came together to function as one super protein, okay? So I hope that makes a little bit of sense. Make sure you take some time to kind of memorize the difference between the primary, secondary, tertiary, and quaternary level and what's going on in each of those. So moving on in these notes then, we have a few more really important things to talk about. So now, let's talk about things that could affect a protein structure. So I told you that the structure of a protein, this final structure and shape and conformation that it has, is vital to what it's doing inside of your cells. And so that shape is going to determine its function, and its function, it can only accomplish its function if it has this specific shape. And if you mess up this shape, even in the slightest, it could have a huge effect on its ability to now accomplish whatever it's supposed to be doing inside of the cell. And so for proteins, their structure is really important because it has a very specific structure to allow it to do a very specific thing inside of your cells, a specific task, or have a specific role or function inside of your cells. And if that's messed up, it's going to affect whether it's able to do that or how well it's able to do that. So there's two main things that could affect a protein structure. One of them is a mutation. And so if you look at the information here, it says a change in just one amino acid in a polypeptide, which is its primary structure. Like let's just say I changed one of those amino acids in that long chain. That could potentially change the structure of the protein and how it's going to end up folding up, which could then affect its function. And so this would happen when you have a mutation. A mutation is when your DNA changes. There's a change in your DNA, and it's your DNA that actually determines or controls the sequence of amino acids. So your DNA has the instructions for what order these amino acids are supposed to be in. And if your DNA is mutated, has a mistake in it, it might mess up the sequence of amino acids that are supposed to be in this certain protein, and that could affect its structure. So that's caused by a mutation. A mutation would cause the amino acids to be incorrect, or one or more of the amino acids to be wrong. So if I go back to this picture here with all of these amino acids, all 127 of them, you could have a mutation that changes one of these, maybe number 25. Instead of alanine, this amino acid, there's a different amino acid that's here instead of this one. And that one change, just changing one of them, could actually, many times actually, very commonly affect how it's going to finally fold into its three-dimensional structure, which could then ruin or negatively affect its function and its ability to do its job inside of your cells. And so this would especially be true. So right here it says a mutation that changes a protein's primary structure, the sequence, can lead to changes in its secondary and tertiary and quaternary structure, how it actually ends up folding into its final conformation, which could ultimately affect its final folding and shape and its ability to function properly. Now, this would especially be true if an amino acid is switched with another amino acid that has different properties. So for example, let's say there's a mutation that leads to a polar amino acid being switched with a non-polar amino acid. So like if I go back to this picture here, if I change, let's say I take one of these amino acids, let's say one of these amino acids is non-polar, and there's a mutation that changes it with a polar amino acid. Now, one of those was hydrophobic and doesn't like water, and one of those is now hydrophilic and does like water. And that's going to change maybe how that area of the amino acids is going to fold up, especially since those hydrophobic and hydrophilic amino acids help with the folding and where certain amino acids are going to end up. And that could definitely, very likely, affect the protein structure. And so here's an example, a real-life example, a very common example with hemoglobin. This is the protein that carries oxygen in your guys' red blood cells. And there's actually a mutation that occurs in humans that leads to this protein being made incorrectly and folding incorrectly, which actually causes a disease called sickle cell anemia. So if you look at this picture here, there's this is showing you some of the DNA that codes for this amino acid. You don't have to worry too much about the DNA stuff. We'll talk about that a lot second semester and how this is happening. But just know, like, there's a certain amino acid sequence in the hemoglobin protein that you're supposed to have. And this is just showing you some of it. There's actually 146 amino acids in this polypeptide. And this is what it's supposed to have, like this sequence here, and then it continues on. And that's going to create hemoglobin proteins that fold correctly, like this shape right here. And when those are found in your red blood cells, you end up with these nice plump looking red blood cells that do a great job at carrying oxygen in your blood. But there's a mutation that some people get or have or inherited where just one of those amino acids, because of your DNA being messed up, one of those amino acids is now switched with a different amino acid, and it's this amino acid right here, glutamic acid, being changed for valine. And when that happens, and that's it, that's the only difference. Out of 146, this is the one difference. And that one difference causes the hemoglobin protein to start looking more like this, which is not the way it's supposed to look, all these hemoglobins. And they start creating these sickle cell shaped red blood cells. Your red blood cells look like these sickle cell shaped red blood cells. And these do not carry oxygen very well, and they clog your veins and your arteries sometimes, and it's not good. And so that creates a certain condition that these people now have, which is not ideal. And so that's because of this protein not folding correctly, and it's not folding correctly because of this mutation, which is affecting its primary structure and then ultimately its secondary, tertiary, and quaternary structure that it's supposed to take on. It might be also important to note that this is not always the case. Not all mutations disrupt the functioning of a protein. So just keep this in mind because these examples come up as well. It is possible to have a mutation that changes an amino acid with a different amino acid that actually does not end up with the protein folding incorrectly, or maybe the protein folds mostly correctly and is able to still work mostly normal, and so it doesn't really have an effect on the individual. And so, for example, if you had a mutation that takes an amino acid with and you change it with one that has similar properties, it might have little to no effect on the shape of the protein and its functioning and how it's folding up. Like if you took a non-polar amino acid and it gets switched with a different amino acid, but that different amino acid is also non-polar, it could be true that that protein ends up folding the same way, because those are very similar amino acids, and really that mutation didn't cause the protein's function to be ruined. So that happens sometimes too. So that's mutations. Now, another thing that could affect the structure of a protein is something called denaturation. And denaturation is when the sequence of amino acids is not affected. So let's say you have a certain sequence of amino acids that folds into a specific shape, and that's fine. That's locked in. Let's say there's not a mutation, so that's the same. But what happens sometimes is or a lot of times is the environmental conditions that protein is exposed to might change in a way that disrupts the hydrogen bonding and the R groups interacting with one another, and causes that polypeptide to start unfolding or folding incorrectly. And we call that denaturation. Denaturation is when a protein, its structure, gets disrupted or ruined because of changes in the environmental conditions. So things like the pH and how acidic its environment is, or the salt concentration, or the temperature and what the temperature is that protein is exposed to, those type of environmental things, they're not going to change the sequence. It's not a mutation. It's not going to change the sequence of amino acids. So the primary structure is not affected. But what's happening is when you change, like temperature, for example, that protein will start to fold incorrectly or start to unfold. And when it does that, that's going to affect its function. And so again, we call that denaturation. So it's not a mutation, but it's still causing the protein to fold incorrectly. And so that's because of these things like acidity or temperature affecting those R groups and the hydrogen bonding that's supposed to happen. And it causes that protein to unfold. And proteins are basically made to fold correctly at a certain condition. So depending on the protein and depending where it's functioning and what type of organism it's in, every protein has a specific temperature and a specific pH and acidity that it's used to. And when you change that protein was made to fold correctly at that specific condition, those specific conditions. And when you change those conditions, almost always that protein will start to fold in an incorrect way and start to misfold and denature. Now, denaturation is actually is usually pretty reversible. If you just return that protein back to its original environmental conditions, like the correct temperature and pH that it's supposed to be in, it usually will fold back to normal. And so sometimes this can be temporary, as long as you return it back to its environmental conditions that it's supposed to be in. And that's because it's not a mutation. So remember, you're not changing the amino acids. The amino acids are all in the correct order. You just need to put them in the right environmental conditions to make them fold correctly. So that's what denaturation is. And again, remember that denaturation, this is affecting the secondary, tertiary, and even the quaternary structure, if there is one, of a protein. But it's not affecting the primary structure. So the primary structure is fine, and that's something different between mutations and denaturation. Mutations are affecting the primary sequence, the primary structure, and everything else. Whereas denaturation, we're just affecting like the secondary, tertiary, and the quaternary structure of a protein, but not the primary structure, the sequence. And so these notes end with, we're finally at the end. Again, I told you this was going to be kind of a long video. There's a lot you need to know about proteins. But at the end here, it kind of summarizes some of the different variety of things that proteins do, the different types of proteins and all the different types of responsibilities they have inside of your cells. So proteins, they do a lot. Some proteins act as enzymes. Enzymes, I pointed this out before. Enzymes, these are the molecules that control chemical reactions taking place inside of your cells. So enzymes, these are actually proteins, and they act as catalysts to speed up chemical reactions that need to take place inside your cells. So when I talk about enzymes, I'm actually talking about proteins. These are proteins that folded into a specific shape, accomplishing the specific task of helping a certain reaction take place. There's also transport proteins. We'll talk about these later on this year. These are proteins usually found in the cell membrane of a cell, and they move things in and out of the cell. So they help things go from inside the cell or outside the cell to the other side of the cell. They can move things across the cell membrane. That's super important. So these are proteins that folded up into a specific shape that are accomplishing this specific function of helping move this specific blue dot into the cell. There's also contractile and motor proteins. These are contractile and motor proteins. These are proteins that are involved in movement. So when there's movement taking place inside of a cell, or on an organism level, an organism moving, that is almost always has to do with proteins. Proteins are responsible for moving things in specific places inside the cell. Inside the cell, there's these motor proteins who can grab onto things and move them to different locations inside the cell. They can also help proteins, or they can actually also help cells like physically move in their environment. And then there's contractile proteins, which are found in your guys' muscle tissue, and these proteins help with contractions. And so they allow like an organism to like move its body. So like me moving my bicep muscle and pulling my arm up like this, that's because of proteins in my arm. My muscle tissue is made of a bunch of these contractile proteins that allow that to happen. So those type of proteins help with movement, cells moving, things moving inside of a cell, organisms moving, all that has to do with proteins. There's also receptor proteins, which are proteins that are found on cells that bind to signaling molecules. So there's certain signaling molecules that bind to receptor proteins. And when they bind to these proteins on the cell, it causes things to happen inside of the cell. And we'll talk about this in Unit 2 a lot. A lot of times those signaling molecules are also proteins. Those signaling molecules, specifically hormones, a lot of your hormones are proteins, which are signaling molecules that bind to receptors in your body on different cells and cause different things to happen in cells and in different parts of your body. And so those receptor proteins, along with these signaling molecules like hormones, which are traveling in your blood throughout your body, those guys are usually proteins. So this is a protein. This could be a protein binding to this receptor, which is a protein causing certain things to now happen inside of your cells. So that's happening all the time in your body. Cells receiving and binding to signaling molecules, and things now happening inside of your cells. And then the last couple examples on here are defense proteins. So there's proteins found in organisms that help defend them against bad guys, invaders, bacteria, viruses, and other pathogens. And a lot of times proteins are involved in that defense. And so, for example, one example is antibodies. You guys might have heard about antibodies, especially after all this COVID stuff. And antibodies are proteins. These are actually proteins found in organisms. For humans, for example, certain organisms like humans, we can make antibodies that are actually going to target specific bad guys. In this case, a virus. Let's say it's the coronavirus. If you have antibodies for the coronavirus because you got the vaccine or because you got infected with COVID earlier, your body has learned how to make these antibodies, which are proteins that target and bind to these viruses and other bad guys in your body. And that allows your body to then destroy it. Those antibodies are going to target this guy to be destroyed inside your body. So it goes away, which is super convenient and helpful. And then very lastly here, we have some proteins that have a structural purpose, kind of like carbohydrates. There's some carbohydrates that build certain structures inside of cells. There's also some proteins, lots of proteins that are building certain structures inside of living things inside of cells. So for example, keratin, that's a protein that forms your hair and nails and the horns of animals. That kind of stuff is made of keratin protein. Collagen, that's another type of protein found in connective tissue, like underneath your skin. There's a bunch of collagen that gives structure to your skin. Ultimately, that's what unfortunately, as you get older, that collagen starts to break down and you start getting wrinkles. But that's a structural protein found in the body of different organisms. And then the cytoskeleton, you might have learned about the cytoskeleton in your freshman bio class, but the cytoskeleton is this whole network of protein fibers inside the cell. So inside the cell, there's this whole network of protein fibers made of proteins that give the cell structure and support inside the cell. So it kind of is like the skeleton of the cell, but that's made of proteins too. So anyway, hopefully from that list, you can see that proteins do a lot. And there's a lot of other things we could talk about. They're doing thousands and thousands of different things that are super important inside of your bodies. But there's a nice little summarized list for you. Anyway, that finally brings us to the end of topic 2. Hopefully you feel pretty good about your knowledge of proteins and your understanding of them. You do have some studying to do, hopefully, to memorize a lot of this stuff and some of these details that we went over. But that's it for this video. Anyway, thank you guys for watching. I'll see you later.